So as I said, please turn off all your gadgets. You know, Georgia Tech has this in technology. When your cell phone rings, it explodes. So it's really a pleasure to introduce our guest, Dr. Michael Levitt from Stanford University. Before I say a couple words about him, I'll tell you about our series. So I'm Tzvi Galil, Dean of the College of Computing, and those of you who have been here long enough probably knows that we have a series of Nobel laureates. Most of them Israelis, by the way, for some strange reason. You know, if you hear my accent, it's not a southern accent. So I arrived in 2010. In 2011, Aaron Chickanover gave us the first lecture, Nobel laureate, and something really funny happened, because when I introduced him, I said that Israel actually had quite a lot of, relatively to its size, of course, Nobel prizes in recent years, after 2000, and probably, I conjecture, and I was happy to be found wrong, that it will be a while before there will be more Nobel Prize winners. And the next morning, the Nobel Prize was announced, and the winner was Dan Shechtman, also from Israel, and he was invited the following year. Then the next year, in 2013, we had a non-Israeli, Ken Arrow, a 91-year-old, very famous economist, also Nobel laureate. And today we have Michael Levitt. So Michael Levitt is not only Israeli, he has 3, maybe 4 nationalities. So I have 2 passports, and I thought that that's a large number. Now, Michael was born in South Africa. He doesn't have a passport. He may issue one. But then at high school, I think he moved to UK, and he's British citizen. Then he spent quite a number of years in the Weizmann Institute in Israel, and he's Israeli and knows. I tested his Hebrew. He passed, and of course he's American. So we decided to give him the Georgia Tech treatment when he came here. So somebody suggested we'll actually give him drown proofing. I'll explain in dinner what it is, except that somebody else said it's illegal. So instead, we put him through 7 to 10 meetings with students and faculty. We made sure he's exhausted enough so we'll get all the information out of him. So it's really a pleasure. And Michael, together with 2 others, one of them Israeli, won the Nobel Prize in Chemistry, though all those that won in chemistry from Israel are not actually chemists and are not in the Department of Chemistry. By the way, another winner was here in Atlanta and Georgia Tech, but not in our series, Ada Yonat. She also won the Nobel Prize in Chemistry. But none of them is, for example, Michael is in structural biology and computer science. So it's a real pleasure to welcome Michael Levitt. At the end, we will have questions and answers, and please use the 2 mics on the 2 sides to ask questions.
Thank you very, very much, Tzvi. It's a real pleasure to be here. You know, there are lots of Nobel Prize winners who are not Israelis. And I think what is really interesting about the USA, the USA has most Nobel Prize winners who were not born here. They have 31% of the Nobel Prize winners were not born here. That's like the biggest either deficit of payment or balance of payment of any other country. I think Switzerland is next with 12%. So, okay. So it's really, really nice to be here. I'm going to speak on this side so I can kind of have some free area. It's really nice to be here. I think this is my 3rd or 4th time at Georgia Tech. I can't remember. And I've had a busy day, probably one of the hardest days in my life, but you underestimate me. I have a lot of stamina, so, you know, I'm back up to shape, it'll be fine. So I'm going to try to give you an overview of a field of macromolecules where I guess I was lucky to be there at the beginning. It's a field that now involves thousands of people, many different advances, and that's the story I want to tell you. Oh. So I'm going to say a little bit about how modern biology began, talk about the birth of computational structural biology, which is sort of where I come in, the future, which is multi-scale dynamics of huge systems, a little bit about applications to human health, and then some general thoughts. Tomorrow I'll be giving a specialized talk on determining structure with less information. So how it all began. And by it, I mean the realization that biology is much more like clockwork than, a soup. I mean, it's very easy to think, you think about inside us, you kind of think about all the blood, but in fact, there's an incredible organization that spans maybe 10 orders of magnitude, which is quite unprecedented. So biology is the ultimate nanomaterial. So I was very lucky because I was, I guess, a teenager in the 1960s, and I was able to essentially stand on the shoulders of giants, which really helps. Linus Pauling, probably the greatest chemist who ever lived, had in the early 50s predicted certain properties of proteins. And prediction is always an important thing because in biology you sort of don't have, it's not like physics where you have a master equation. Biology is evolution, it's subject to accidents and things like that. So predictions are hard. He predicted the alpha helix, which is an area where the polypeptide chain winds on itself, in 1951. It wasn't really observed in proteins until about 10 years later. The other person I think is probably one of the most familiar characters, Francis Crick. He is most well known for DNA. He actually did the DNA during his PhD thesis, and the DNA paper is just stuck in the back sleeve of his PhD thesis as, like an afterthought. He again, using sort of logic and a little experimental information, was able to predict a structure which had amazing importance. But perhaps the person who really changed the whole view of biology was actually this man, John Kendrew, and he certainly had more influence on me than anybody else. He was the person who determined the first three-dimensional structure of a protein. This is actually sperm whale myoglobin. They used whales because I guess this would be totally illegal today, but it was very easy to get large quantities of whale muscle. And this is a protein that actually stores oxygen in the muscles of whales. Whales have to dive very deeply and their lungs collapse, so oxygen is stored in their muscles. This is a protein which consists of something like maybe 1,500 atoms. Here is a heme group. And what's critical about this is this molecule has a function that depends on its shape. The shape is very precise. Here's an alpha helix, another one, so Pauling is verified. This is the first protein X-ray structure. Today there are about 100,000 such structures, not totally different, but basically this brought forth the paradigm that biology works by structure, both in time and space, leading to function. And this, at least for me, was a total revolution. About at this time, so it turned out that Kendrew published his work in Scientific American. And the reason he published it in Scientific American, it was the only place where you could get colored pictures. So in the 60s, I was 14 in 1961. At that time, it was great to subscribe to Scientific American because you were picking up this like first-hand research from these, really people, and they just wanted to publish there because the colored pictures, and they were very, very good articles. In those days, there was no molecular graphics. This was actually made by a painter, Irvin Geiss. Okay. The next person is maybe a little bit less well-known, Max Perutz, but he really probably had more influence on the field than anybody else. I think this is his picture from Scientific American. He wasn't able to get atomic resolution. In Kendrew's structure, you can see all the atoms. Perutz was able to just see that essentially hemoglobin in our blood is essentially 4 myoglobins connected together. So nature So this immediately shows that what it's used from whales in one way, it's reusing for human beings. It stores muscle, it stores oxygen in the muscle of whales. It transports oxygen in the blood of humans. Perutz, for me, was the real hero. And the reason is that he was actually the PhD supervisor, both of Kendrew and of Crick. But he didn't put his name on any of their papers. And this was actually quite common in Cambridge in those days. So it was like, he was just a great nurturer. And the fourth person I want to mention doesn't have a Nobel Prize, David Phillips. He managed to make the cover of Scientific American in 1966 with the first three-dimensional structure of an enzyme. By this time, two structures were known. So we had one structure in 61, another one in 66, and as I said, there are 100,000 today. So now lysozyme is an enzyme. It carries out a chemical reaction. It immediately raised a question of how enzymes carry out reactions, and part of my story deals with that. David Phillips actually died relatively young and was a very important supporter of computational biology. Okay, so how do I get into the story? So essentially, one realizes in any career that luck plays an enormous part. Just happening to be in the right place at the right time. And in some senses, by being born in South Africa, there was one thing about being born in South Africa. There was no television in South Africa at that time at all. And at the age of 16, I came to visit my aunt and uncle in London. They had a black and yellow television, probably a really expensive one. And I was totally addicted to television. So besides watching the Winter Olympics of 1964, I watched a program that John Kendrew had made. It actually aired on the 4th of January, 1964. And the titles here could be from a modern course on biological structure, proteins in one dimension, proteins in three dimensions, reproduction and genetics, messenger of the genes, solving the code, living architecture for viruses, etc. Now, what's remarkable about the series is that John Kendrew won the Nobel Prize at the end of 1962 and immediately sat down and made the series. So that's kind of amazing. And I've now heard from probably 3 or 4 Nobel Prize winners that said that they went into structural biology and molecular biology because of his series. Very sadly, the BBC couldn't keep the tapes. They had to reuse the tapes, so this series does not exist. And that's too bad. They still sell his little monograph on Amazon. Okay, so after seeing this in 1964, I started my degree at King's College in London, I guess at the beginning of, in October 64. Reached the end of my degree 3 years later and wanted to do a PhD with John Kendrew in Cambridge. I wrote to him, he said, sorry, we're not taking any PhD students this year. So I said, okay. But I had friends who were in business school. And they said to me, if somebody says no, you've got to come back with a counter offer. So they said, why don't you just tell him, what about the next year? And I think at that point they realized that this could be inductive. And therefore they said, come for an interview. I went for an interview. I met Kendrew, I met Perutz. The interview went very well. They said, okay, great interview. We'll tell you in a year's time whether we'll accept you in a year's time. I said, fine. My friend said, are you crazy? I mean, you're going to do nothing for a year. You're going to just sit around. Which actually sounds like a pretty good idea now, but at that time, doing nothing for a year didn't seem like a good idea. So you should go back and tell them that, what's really happening. So probably one of the most brave things I ever did is I drove up to Cambridge in April of 1967, happened to stood outside Max Perutz's office until he came towards his office, got hold of him in the corridor and basically said, I'm terribly nervous. I have my final exams coming up. I can't study because I don't know what's happening about my future. It seems like a logical story. He said, very embarrassedly, come into my office. And he quickly said to me, look, I can't help you. John Kendrew and I are both joint heads of this place. I have to talk to John Kendrew. So I immediately, very embarrassedly, left. And what happened was by Monday, they decided actually to accept me in a year's time, subject to one condition. I had to spend the year before I came to Cambridge in Israel. So in 1967, John Kendrew sent me to Israel. It's a time when many Jewish people actually went to Israel. It was after the 6 Days War, so everyone assumed I was a volunteer. But he was sort of sending me, I wouldn't say against my will, but I really prefer to go to maybe Boston or Tokyo or Paris or something like that. But he said, no. And then he did something that was kind of remarkable. They had just established a postdoctoral fellowship series between Israel and England. And because it had just been established, there weren't any candidates. So I said, okay, you'll go to Israel, we'll give you a postdoctoral fellowship. This was a huge amount of money. It was like 4 times more money than I'd been making as a student. So I was kind of bribed to go to Israel. Anyway, I went to Israel, and the reason Kendra had sent me to Israel, he was actually on the board of governors of the Weizmann Institute, and he knew very well the work that was being done by Shneur Lifson. And Shneur died 13 years ago. But basically, this is probably 3 or 4 years before then. This is how Shneur looked when I came, and this is how I looked. So kind of a cool 60s picture. Okay, so why did Kendra want to send me to work with Lifson? And, it's very funny because it's only in giving this talk that I started to realize how he knew it was going to happen. I mean, the reason is that he realized that biomolecules are detailed. There's the pictures from Scientific American, myoglobe and lysosome. They look different because they do different things, but they're made of exactly the same components. And essentially, these come from crystals. So in the crystal, you're looking at a million molecules, and they all look the same. So basically, structure is extremely detailed. Atoms are precisely positioned to within 1/10 of a nanometer, very precise. So there must be forces which could actually understand how this is. Now, Lifson wasn't working on proteins. He was working with Aryeh Varshil, who also won the prize, on alkanes. These are molecules made up of CH2. And he said that you needed to have a philosophy, and his philosophy was called the consistent force field. So a force field is a set of interactions and forces between atoms that determine the system. Trouble with force fields are you don't know what parameters to use. You don't know how big the atom should be. You don't know how long the bonds should be. You don't know how stiff the bond should be. And at that time, people who were doing measurements of these things by spectroscopy kind of used whatever parameters fitted the spectroscopy best and didn't worry about a CH2 group always being the same entity. Lifson said a CH2 group, wherever it occurs, whether it occurs in a cyclic molecule like cyclohexane or a linear molecule like a hexane, should actually be the same energy parameters. And my job when I got to Israel, this paper came out at the end of 68, I got to Israel in October 67, was basically to write a computer program that, given any molecule, would calculate the structure, the minimum structure, the energy, the second derivatives, and the vibrational spectra. I had never written computer programs before. I was handed the Fortran 2 manual and told to write it. I still can't believe that like 4 months later I had a working program. The good thing was the computer was actually in the Weizmann Institute, and you could actually feed your cards in as often. If you stood by the card reader, you could get the jobs run pretty often. So it wasn't the normal 1 turnaround a day. I might have got 4 turnarounds a day and so on. Anyway, so with that program, they were able to calculate these properties of molecules and then do least squares to fit the experimental values by this method. Okay, so the energy function of a molecule at that time looked like this. Essentially, you have a potential energy, which is the sum of many terms. For example, every bond is a spring, i.e., this is a forceful law for a spring. B is the bond length. B0 and Kb are force constants, parameters. If you stretch the bond, the energy goes up. If you compress the bond, the energy goes up, and it's a spring. Bond angles are the same. Here are van der Waals interactions. 2 atoms can't interpenetrate. If they get very close, the energy goes up, like this. If they're very far apart, they don't interact. And in between, every pair of atoms attracts each other. And then you have Coulombic interactions where a positive and negative attract each other, and 2 like charges repel each other. And then you also have torsional terms for twisting about single bonds. Now, this is a very, very simplified representation. The energy is written as a sum of these terms. And this makes it relatively easy to calculate the energy. Essentially, in here, there are things which are variables. R is the distance between 2 atoms. Phi is the torsion angle. B and theta, all the other terms are parameters. So the idea was to essentially determine these parameters by least squares by fitting the property of alkanes. Okay, and that basically worked pretty well. Now, one really important thing is I chose to study physics because I felt that I didn't want to learn very much. And in physics, you can sort of get many things from first principles, at least for simple classical physics. One of the really important principles is if you have the energy, potential energy as a function of the coordinates, you can calculate all sorts of things. So here's an example, or one last thing. If you put dates on this, you can end up calling this the dead physicist's potential. And I make that point because what is rather surprising is this particular energy function is evaluated probably by half the supercomputer power in the world. And it's a bit crazy because this is an old-fashioned energy function. So one of the points here is going to be we need better energy functions. But for whatever reason, in the last 50 years, things have hardly changed. Okay. So if you have an energy function, you can do cool things. Here is a one-dimensional example, position energy, a bit like a roller coaster. So one of the easiest things is to do minimization. You start somewhere, and you drop into a local minimum. That requires generally the forces. So you need to calculate the forces to do that, and you just follow the forces downhill. There are a number of different methods. Another method is normal mode dynamics, where you vibrate about a minimum. This is actually harder. For this, you actually need to calculate the second derivative, the curvature of the surface, and then analytically you can calculate the modes. Molecular dynamics essentially is using the same force that energy minimization uses, but keeps an account of the momentum, the kinetic energy, and then you can actually move over barriers and explore the surface much, much better. And again, what works in one dimension in physics usually works in n dimensions. And then finally, we have the Monte Carlo method, where you jump about randomly. We can put dates on this, Euclid to Metropolis. Now, Metropolis is really interesting, because this is the dates of birth. Metropolis's paper came out in 1955. Metropolis, Rosenbluth, and Teller. Teller being the father of the hydrogen bomb. So this method was a classified method that had been used to study neutron diffusion in the hydrogen bomb. Okay. Now, at the same time I was in Israel, a biochemist named Nathan Sharon, who was working on lysozyme, had written to David Phillips, his experimental colleague, and asked him to send him the coordinates on a printout so he could build a model of the structure. Now, he didn't really want to build a model, and he had a PhD student, Yuval Ashtet, who also didn't really want to build a model, but together we built the model, and I was involved in building it. Building a model is something which hardly anyone does these days. It is a pain. It's like a three-dimensional jigsaw puzzle where things don't really fit together, but you've got to measure them with a plumb line and coordinate paper. The good thing is at the end, you really know the structure well. But in my case, this was actually an opportunity to put this into the computer. The reason was in those days, you couldn't get coordinates. But here I had a listing with all the coordinates. Without asking anybody, I took the coordinates over to the typing pool, and they typed them onto punch cards. And I could then take lysozyme, and I subsequently got myoglobin and put both of these structures into the computer. So this led to my first paper on protein energy minimization. So in some ways, taking Lifson's idea of consistency very far, I said if he had energy parameters for hydrogen and carbon, I could guess energy parameters for all the others. And it's not as crazy as it seems, because you have single bonds, which are less stiff than double bonds. Oxygen is typically bigger than nitrogen. A lot of this was done. There wasn't really enough experimental evidence to tie these things down. The important thing is that I was able to express the potential energy and very easily realized that the same computer codes that could calculate first and second derivatives of small molecules could calculate just the first derivatives of large molecules, because the second derivative required a matrix, and that required the same space as a much bigger molecule as a vector. So I guess we were using memories then of a few hundred kilobytes, maybe 200, 250 kilobytes. We could actually take, in this case, the protein lysozyme and myoglobin and minimize the energy doing something like 50 iterations. This took about, I think, 18 minutes, the paper says. So we could have done much more. But the important thing was the structure came down to low energy, and we had done the first calculations of forces on a molecule. I then finished my year in Israel. Meanwhile, I got married to an Israeli, went to Cambridge, finished my PhD there, I guess my wife had the two children, and I don't know how much help I was, but I hate to think what I was like then. We're still married, so I guess she doesn't give up. I went back to Israel and worked on multi-scale modeling of macromolecules. So multi-scale modeling is something I never actually used as a term. We just called it, simplified, more complicated, and so on. But I guess this is the term that was used by the Nobel Committee, and they were very excited about this term. I'll tell you at the end why. So what is it? So you can, blame it on Einstein. Einstein is quoted as having said that everything should be made as simple as it can be, but not simpler. It turns out, apparently, that it wasn't actually said by him. It was said by a newspaper reporter, Roger Sessions, who wanted it to sound more important and said that Einstein had said it. But I mean, in some ways, this is just common sense, except it's really important when it comes to calculations. Because basically, if you do a calculation that is incredibly complicated, you can't do it. And if you do a calculation that's too simple, it wasn't worth doing in the first place. So somewhere in between, there's like a golden position, a sweet spot, where the calculation is complicated enough to be worth doing, but simple enough to actually get the details. And in some ways, I think we managed to find 3 such approximations, and that was important. Okay, so the first thing we did was coarse-grained models. We just called them reduced or simplified models, but I guess coarse-graining is a well-known term in mathematics that I was completely unaware of. And essentially, the idea was to take a polypeptide chain and basically leave out many of the atoms, and then be able to fold up a simplified chain by combining energy minimization, which we could now do 1,000 steps of in a few minutes, with a method to use the normal modes as a way to jump out of the minimum. It turns out we weren't actually calculating second derivatives, but in those days people had started to invent methods. People like Fletcher and Powell had invented, and Davidon had invented variable metric methods, which created an approximation to the inverse Hessian during the minimization. You could just diagonalize that and get normal modes out of that. So it was a quite complicated method, and it was able to fold an extended chain into a compact shape, which sort of looked a little protein-like. Anyway, but the hard part here, it turns out that nowadays you would think nothing of leaving out atoms. But in those days, we thought that atoms are holy. You don't just leave atoms out. So it's interesting to think how this came about, and I remember it very clearly. In the early 70s, and this was started in 73, in the early 70s, space travel was quite common. The Apollo missions were going up into space and so on. And we thought, wouldn't it be nice to take heavy plastic models into a space shuttle and play with them as if we were doing protein folding? Okay, well, of course, we couldn't do that. So then we said, let's simulate that process, i.e. a molecule without gravity as a model. We're not simulating real molecules, we're simulating plastic models. So we sort of, in our head, made a plastic model like this and played with it in the computer. And this allowed us to leave out all the atoms and do this. It turns out that, surprisingly, as computers became more and more powerful, these reduced models, these coarse-grained models have become more and more important and are generally used in many methods of simulating protein folding, looking at large systems, etc. At the same time, we did something else, which is called QMMM for quantum mechanics, molecular mechanics, and we again did not invent the term, but it became adopted as a term. So this was to study catalysis. So when Phillips published his paper 10 years before, in 1966, he hypothesized how lysozyme works as an enzyme. Lysozyme cuts a sugar molecule in the outer wall of a bacteria. And he thought it does it by binding the sugar and then sort of pulling at the ends and breaking it, applying steric strain to the molecule. I had tried this during my PhD and come to the conclusion that the enzyme was too soft for this to happen. You couldn't, it's like trying to break somebody's back on a nice soft bed. It's not so easy to do. A hard floor is much easier, or a rack, so you can't really exert enormous forces on a nice soft enzyme. So how did it work? We had hypothesized that it actually worked progressive electrostatics, but that needed to be calculated. So here is Aria Varshal, about at that time. Here is just a very small part of lysozyme. This is, in fact, a 6-membered sugar ring. It's a chain of such sugar rings. It carries on down here. Here is a glutamic acid inside the protein, a COO group with a proton here, which probably should be deprotonated, but I'm not sure. And the idea is how does the orbital, so now what you do is instead of making things simple, you add in all the electrons and use quantum mechanics to deal with literally a handful of atoms. Of course, you have to have the rest of the structure there, both to hold the bits that you're having, but also if you're generating electrostatic fields and changing them, those fields have an influence on the rest of the protein. Equally well, the rest of the protein has an influence on the electrostatic field. We put, in this paper, we actually introduced a very simple model for solvent, and that was now the second part. Okay. At this point, the word started to spread. A very famous paper involving Andy McCammon, Bruce Gellin, and Martin Karplus, who was a co-winner, introduced the dynamics of folded proteins. They were doing calculations that were very much based on work of Anna Suraman, who had early on done simulations of dynamics of argon and then water, and these water simulations very much set the way. And in doing the simulation, they had run the system for something like, I think, 10,000 time steps or 15,000 time steps, and things move. If you look really carefully, this is at time 0, and this is after, I think, 3,000 time steps. The end of the molecule is moved. But of course, that isn't, how do you look at a moving object? And the obvious answer is you make a movie. I actually did this together with Richard Feldman at NIH in the, I think it was 78. I sent him coordinates, and he basically made this movie. We now actually have all the hydrogen atoms which were left out, and we actually have some water molecules in the system. And if you actually ask, this movement looks kind of strange. I've tried to say what it looks like. Maybe it looks like people dancing in a disco that's got a very funny shape and you're viewing it from above. Everyone's moving, but no one's actually going anywhere. Or maybe a bag of worms. But this is what molecular dynamics looks like. And it's because things are juggling around. It's also clear that the van der Waals interactions are very dominant. There's also a water molecule that sort of orbits out of the system because there aren't water molecules in the calculation. Putting water molecules in the calculation was actually very hard. Raman had shown that you could simulate water, but the force field he was using was very different from the force field that was being used for, in detail, was very different from the force field used for the protein. So I spent the next, I guess, almost 10 years trying to do this. I was actually at the Weizmann Institute then, and together with Ruth Sharon, an engineer there, we were able in, I think, 86 to get 200 hours of Cray XMP time through the courtesy of the NSF. This is like $200,000 of money back at that time, and I'd used that money to run this calculation. It was a big calculation. The reason is that when you add the waters, you have to put the hydrogen atoms in, and the calculation takes about 100 times longer. And why would you do it that way? The reason is that this is a measurement. Here is time in picoseconds. This is distance from the starting structure. And basically, in vacuum, it moves a lot further from the starting structure than it does in water. Basically, because in vacuum, in the crystal, there are water molecules which are very important. In pure vacuum, proteins have a different shape than they have in water. So subsequently, almost all calculations on protein dynamics are done in water. Here's a movie of some protein dynamics. This is a small alpha helix in water. This is a movie that was actually made back in 1990. It was made by taking a camera and sticking it in front of the screen. I should have realized that you want to align this really, really carefully. A good photographer would never have made such a terrible alignment. And this just shows an alpha helix, the structure that Pauling had predicted, moving at normal temperature in a bath of water. The waters are still here. You can see hydrogen bonds breaking. Nothing much is happening, but you wouldn't expect much to happen at this temperature. So what we're now going to do in the next sequence is bump the temperature up. Here we're going to look at unfolding at very high temperature, and at high temperature, the water molecules are moving very quickly, and the structure unfolds. Now that is like common sense, but actually common sense is a very important test on calculations. So that was an important thing. I was quite lucky because I gave a lecture where Pauling attended in 1990, and we actually made these movies for him. The last scene is actually interesting because it shows a space-filling structure moving. Yeah, so it's kind of... The atoms seem to disappear because it's the way we take out the high-frequency motion by averaging frames. Okay. At the same time, we actually ran simulations of DNA in water, which was much harder, because DNA, here's just a base pair in water, just a GC base pair in water. It starts out paired, but it stacks, and it was sort of known in those days that DNA prefers to stack than be base paired in water. It's base pairs in the DNA because it's a whole stack, and it can't do that. Here we can actually see a double helix in water. This, again, is quite a big calculation for 1990. And here we see DNA with its very charged, negatively charged phosphate groups. There are counter ions in the system. We'll see them more clearly when we get, so these pink things are counter ions. And basically running the dynamics, not very much happens, and that's what you want. It was very difficult to make the calculation stable because the structure wanted to blow itself apart, and in this case, we could do that. I'm going to skip the next slide. Okay. So now, what about the future? I'm just going to show some examples of what we're currently doing. The real molecules that are interesting now in biology are maybe 100 times bigger than things we're seeing, and they have moving parts. Large chunks move. So when you have moving parts, the way to simplify them, this is a general scheme, is explained here, reduce the number of degrees of freedom. So, when I move my arm like this, I've just moved 10 to the 25 atoms, and every atom has 3 degrees of freedom. So I've moved a lot of objects. But I can basically ignore all that and just think about my arm moving about a few pivot points. So that's a very important principle. Here is a movie of RNA polymerase, an important enzyme in our body made by an ex-postdoc and his student, so he's kind of like a scientific grandson. It's a very nice movie. It shows a system with almost 500,000 atoms moving by a method called Markov state dynamics. It allows you to take, essentially you explore the energy surface of the whole structure and then take large steps. The time is, it would be very difficult to simulate timescales like this. But it's a very pretty movie because it just shows in detail how an object as large as this actually works. RNA polymerase is meant to copy RNA shown here in red from DNA shown here in blue. And the key thing we're going to see is that this nucleotide over here is going to move across to here, and an incoming nucleotide can then increase the length of the stack. And this actually is difficult. It takes quite a lot of time. And it just shows that nature really goes down to tiny little details. So here it continues. This is the cheat sheet. When this starts to move, it's going to come across down to here, goes halfway, it sticks to a phenylalanine ring, and then goes all the way. And then in reality, at this point, an incoming nucleotide would come in here and join the stack. Okay. Another example is the entire ribosome, structure that was rewarded a Nobel Prize. In fact, Roger Kornberg got the Nobel Prize for RNA polymerase. So now we're doing normal modes. We're going back to that second technique and taking very, very large structures. This structure has 50 chains and about, in this case, 200,000 atoms. In this case, we're using a coarse-grained model, and we're just calculating the normal modes. And these modes, which are calculated in about an hour for both systems on my laptop, show a lot of large-scale motion that may be functionally relevant. The key thing is to interact with experimentalists to see what they like about the motion. I'm going to probably skip this one and the next one. Okay. So let me now just say something about applications to human health. Because, the calculations have been expected to design drugs greatly in the last probably 30 years. Hasn't quite happened yet, although there are anecdotal stories of drugs designed principally by computation. A drug is a molecule that binds very strongly to a protein, so in principle, if you could calculate the energies accurately, you could design drugs. But my story is a little bit different, and it basically is a complicated story. I am in a medical school, and about 4 years ago, the dean of the medical school wanted to give me an endowed chair. And he had an endowed chair for cancer research as the next available one. And he said, okay, you are now, professor of cancer research. Now, I had to go and thank the family for this. And I'm thinking, what am I going to tell them? I don't want to change my whole career. It turns out that an endowed chair basically pays your salary. So it's like getting an extra NIH grant. I'm very trying to think I'm going to promise I will work on cancer for the rest of my life. But then I suddenly realized that I'd actually done stuff in the past. So a little bit of repurposing could get me out of the obligation very easily. So the work I'd done in the 80s involved antibodies. So antibodies are proteins. Each of these little lumps are something like 1,000 atoms, very precisely ordered. They're actually quite similar to each other. And the key thing is that on this face of the antibody, it looks like this, And these loops here, 3 from one chain and 3 from the other chain, actually define a binding site. So antibodies bind anything. They're able to bind hundreds of thousands of molecules. It's our immune system. We can recognize friend from foe due to antibodies. And just showing it in molecular terms, antibodies have these faces that can bind different molecules because of where the atoms are. So this led immediately to an idea, not our idea, that you could use antibodies to treat cancer. You could take a molecule that's important in cancer, inject it into mice, raise antibodies. These antibodies would now, in some ways, eliminate that particular bad molecule and treat cancer. So before going into this, I should say that I had, for purely academic reasons, developed a computer program that allowed one to model where these loops should be. And involved a number of papers with lots of other collaborators showing that we could do this. It involved experimentalists and other modelers who modeled by hand, another paper, and we were able to more or less predict where these loops should be. Now the important thing was I had a computer program which, given any antibody, could build a model. It wasn't a great model, but it was an okay model. At this point, I'd moved to Stanford in 1987. There was a startup company there called Protein Design Lab. Interesting founders, Carrie Queen, a mathematician, Lawrence Korn, a geneticist, and they had a very broad focus on protein engineering. They had an amazing scientific advisory board, Roger Kornberg, Levitt, Lee Hood, and Stan Falco. These names are not known to computer scientists, but these are the greats of biology. I was also part of that group, and the idea was to do, it was very unfocused, except that they managed to get us to meet every month. And I don't think I've ever been in an SAB that ever met more than once in the lifetime of the company, but got us to meet, and very quickly, the idea moved, instead of doing protein engineering, we would focus on antibodies. This led quite soon to a paper, a humanized antibody that binds to the interleukin-2 receptor. It involved a person at NIH who had actually had the receptor, other biologists, and somewhere in here, I am there because I'd actually helped them do the modeling. Now, they were clever. People had previously had ideas of how to, I'm going backwards, model antibodies. Greg Winter and others in Cambridge in the mid-'80s, maybe 3 or 4 years before that, had said, what you need to do is take an antibody from a mouse, sequence it, and then make a human antibody, a humanized antibody, by taking most of the structure from humans and adding a small part from mice. Now, Queen had a better idea. He said you should take the red part from the mouse, the white part from human, but if there are any amino acids in the white part that are very close to the red part, they should be candidates for change. And he then coded this in a patent. He said, basically, in order to humanize, it isn't enough to simply transfer the CDRs from the mouse and take an antibody from humans. You also have to change at least 3 amino acids in the blue region. And furthermore, there are many different antibodies in people. And some human antibodies just happen to be very close to mouse antibodies. So it's really a good idea to choose a human antibody which is very close to the mouse antibody, then change a few of these amino acids, and then do it. And they actually spend some time protecting it by patents. The patents expire, I think, next month, but that's 25 years afterwards. But they basically patented carefully and then defended their patents vigorously. And this attracted big biotech and big money, because without patenting, no one is going to invest a lot of money in production and developing this, because it's, and production of these things is very expensive. This was done by the company Genentech, who was later became Roche, and they probably spent between 10 and $20 billion on manufacturing antibodies. They licensed the design principles from Protein Design Lab for a license fee of 4%. And basically, so now what then happened was they worked very hard on developing these antibodies. Meanwhile, Protein Design Lab did really well until 2000, reached a peak market valuation of about $7 billion, and then basically didn't do very well. Right now, Protein Design still exists, but it is 7 employees who are all lawyers living next to Lake Tahoe, living in Nevada to avoid sales tax. And what they're doing is they're collecting royalties, because the royalties are quite massive. For example, in 2012, were bringing in $400 million of royalties. Now, this means that the company was probably making, at least 25 times that from this drug. And these drugs are quite well-known anti-cancer drugs, Herceptin, Avastin. I kind of hope that you guys have never heard of this because it's usually not a good sign. But these drugs actually do help. The patent is expiring very soon. I keep on forgetting to update this. It should be 1 month. I didn't have any share of this, so I'm not any particularly sorry. But I do think that there's going to be a lot of antibody work in the future to develop new drugs. When we were doing this, we said, antibodies are no good. They're not pills. You have to inject them. It's a big drag. But it actually turns out that people who have to take pills forget to take them. And if a nurse comes around every week and injects you, you don't forget. So there's something good about that. In any case, the moral of the story is it takes a very, very long time to go from science to medical value. This is actually very important for medical schools. They are always talking about translational research. And what they want you to do is say, okay, I will solve this disease in 3 years. And I try to tell people that long-term science is like long-term investing. If I say I'm going to invest my money for the next 3 years, I'll be lucky to get 3%. But if I say you can have it for 20 years, I'm sure I could get 10%. So I just think that we need to realize that these things happen, they take a long time, and a lot of serendipity is involved. It was very important. People say, well, isn't it a pity this was patented? It shouldn't have been put in the public domain. No, because it was patented, it was worth a company developing it. It would never have happened. So it's kind of interesting. Okay. Let me just end up with some general thoughts. The first of which is, it's clear that this method, this work, has been pushed ahead by technology. There's no doubt that if computers had not changed the way they have, this would not be recognized. It would be still very much backwater. And this is an interesting thought because a few days after my life suddenly changed, I was asked by somebody, how much have computers changed? And I guess I was actually asked to think. And thinking, up till then I'd just been giving stupid interviews and things like this without thinking. So I sort of sat down and instead of just saying, Moore's law, leave me alone, 10 to the 9, 10 to the 12, whatever you want. I actually sat down, took my laptop and ran the same calculations that I'd run in 1968 on my laptop. And to my surprise, and maybe there's a reason for this, everything seemed to change by 10,000 times. The cost had dropped 10,000 times. The speed had increased 10,000 times. Memory had gone up 10,000 times. Size, I guess it's not clear how many laptops you can fit into a hall, but let's just say 10,000. There's a number. So it's interesting, and some of these numbers really can be multiplied. You can multiply cost per dollar. Dollars per CPU and multiply these. But it looks like we're getting something like 10 to the 9, and this is before I'm just comparing my laptop. I'm not talking about GPUs. I'm not talking about special processors, etc. Okay. So, the obligatory joke is to say that if cars were like a new Volvo, they would cost $3. They would have a top speed of 1 million kilometers an hour. So this was Sweden. They would carry 50,000 adults in comfort, and they would park in a shoebox. Now, that's a joke, but that's true. In other words, everything I've said here is actually true. Prices have come down by 10,000 times. Speed has gone up, etc. And this is a reality which I think completely changed the picture. When you get factors like this, they change things in a qualitative way. What is also important here is if someone had asked me back in 67, would this happen, I would never have predicted this. But if you ask me now, what do I think is going to happen? I feel very confident, because I won't live, saying in the next 50 years we're going to see at least similar kinds of changes, because computing is really happening in an amazing way. Not Moore's law, but putting together processes, etc. So, okay. So just a little bit about what's next. So I said nasty things about the energy functions being used, because I do actually think that you can use much better energy functions. One thing that's happened for all this computer power is that it's possible to do pretty good quantum mechanical calculations on certain kinds of molecules. I can take a methane molecule and a water molecule, and for any particular position I put them, use a program to get a pretty good energy value of the energy of interaction. You can then take that data, and instead of fitting experimental data, you can fit energy parameters to that data. Now, it turns out that this is work that was done by a company called Algodyne. It was actually founded by the lead author of the PDL patent, Carrie Queen. He had made like $100 million from that company. And he decided that one way to get better drugs was to get better physics. And he wanted to get better physicists. And at that time, the place you could get physicists very cheaply was in Moscow. So he founded this company in Moscow in 2001. I was asked to sort of be the SAB. And it was sort of interesting because instead of using quantum chemists, they used quantum physicists, most of whom had never heard of a hydrogen bond, but were very familiar with a hydrogen bomb. And they worked together. It was kind of tough. And they put together an energy function, which is a lot more complicated than, I mean, if you like, it's probably a circa 1945 energy function with terms like exchange and induction and dispersion, etc. Put together this energy function. And the idea is to parameterize this based on small molecule work. Unfortunately, after about 7 or 8 years, Carrie Queen had spent $10 million. And his wife had another company which had basically spent almost nothing. It was making money. So he stopped funding this company. The company is actually continuing in a different guise, the technology anyway. So there's now another company. Surprisingly, this kind of work cannot be funded publicly. No one is interested in any new energy functions. There are too many people who are involved with CHARM or ALMBA or NAMD to want to have any competition. So they're still very happy to use the dead physicist potential function that might be very, very good for proteins and for DNA. But if you think about drugs, drugs are different chemical entities. And proteins are repeating. Once you have the parameters, you can do it. We need to get much better energy functions for drug design. We actually did some calculations involving 2 postdocs, Gaurav and Leonid, where we took a Buckminster fullerene, which is a sort of Death Star-looking molecule of 60 carbons in a shape, I guess, I forget the name, it's a truncated dodecahedron or something like that. Put it in a big box of water, calculated it, and found that it was quite different from what you'd get in classical methods. So this is a measure of how ordered the water molecules are in the surface layer. It's not really important. This is quantum, this is classical, and they look different. The quantum takes about 10 times or maybe 20 times longer. So I think that's going to be very important. More specifically, my current group, it's quite small. I actually have now 4 people, but we're actually doing lots of interesting things. And Andrea is working on comparing whole genomes. Jana Goffman is working on the cryo-EM of membrane proteins. And Ivan is working on how to solve X-ray structures, maybe solve the phase problem, but I think that's maybe unclear. So now sort of stepping back a bit further, I think any scientist who is going to really devote himself to science or herself needs family support. I've been very, very lucky. Here's my mother, who's actually still alive. She's 99 here. Actually, she's 98 there. Yeah. Maybe 99, I forget now. But she is now going to be 100 in March. My wife and my eldest granddaughter. This is not a skin disease. It's the festival of Purim, and everyone's meant to be dressed up. So my wife has painted her face. But my wife particularly has been an amazing source of support. I cannot even imagine what I was like when I was 20. I was probably the most intolerable person. I've actually got better. So, I mean, I must have been totally hyper, totally self-centered. Okay. And, of course, you know the old saying, behind every successful man, there is a surprise wife. This was actually sent to us by a Swedish colleague. Initially, I felt rather bad that I was, this is not a gender-neutral remark. So I tried saying, behind every successful person, there's a surprise spouse. Doesn't work. The symmetry here cannot be made symmetrical. I think, and if you made it behind every successful woman, there's a surprised man, it would just sound really nasty. So, okay. I think in one's career, you need to take chances and make mistakes. Rather than talk about scientific mistakes, I actually started hiking when I was 60, 7 years ago. This is me 2 years ago hiking through a national park in Chile, Patagonia, Torres del Paine. And you might think I'm checking my email, but I'm desperately looking at the GPS to know how much further to go. We're climbing over a very hard path. You'll notice that all the young people, first they're looking at me really weirdly, but they're still wearing their anoraks because it's really, really cold. I've been I'm overheating like crazy, and it's okay. But don't be too stupid. So a little bit after that, I did something which would be perhaps considered stupid. I wanted to go to have a conference. I wanted to have a conference in Sweden. At that time, there was a Sweden-Stanford exchange group. This asymmetry here is Sweden, Stanford, country versus university. But a lot of Swedish people wanted to come to Stanford in the wintertime. And then we decided we would have a meeting in Sweden, so the Americans from Stanford would actually come to Sweden. And I was hoping they'd have the meeting on the islands, hot tubs, saunas, and all of this. But they had it at Uppsala University. So I decided that I was not going to accept that. The weekend before the meeting, I would go by myself to the archipelago. I was going to hike, but one of my Swedish friends said, "You can't hike on the archipelago. You have to walk on water. There's no hills. You have to sea kayak." So I'd never sea kayak before. But I went online and I found a 6-minute flash movie that taught me everything you could ever want to know about sea kayaking. So I went there. I hired a sea kayak. I didn't realize that in Sweden, there's a step function of the occupancy of the islands. Before Midsummer's Day, there is nobody there. And after Midsummer's Day, it's completely crowded. I was there the weekend before Midsummer's Day. I went to the place. I got the sea kayak. It was amazing. I had my camping equipment. I went from one island up to another, found an island over here where I stayed, went into a cove, etc., and came back. It was amazing. The island was really, really beautiful. This is my island. Here is my tent. I actually had the GPS, cell phones work really well in Sweden. So the whole time I had cellular communication. I was talking to my wife by Skype, from the tent and doing my emails and things like that. But I was completely alone. In the time, I think I saw 2 sailing boats, 2 motor taxis, and an army destroyer. So it probably was quite dangerous. Although I guess if you get through it, it's okay. I went back to Sweden this summer and went back to the same places with a friend who had a sailing boat. It was also nice, but it was kind of boring. So I kind of still like sea kayaking. Okay, so the obligatory advice to the young, be passionate. Do something you actually want to do. Because if you do something you don't want to do, you might be successful, and you have to do it. It's not a good idea. Be persistent. I was very lucky that my friend said, if they don't want you, just come back with a solution. I think you have to be persistent, but in a creative way. If I just made it an inductive thing, they'd get bored of me very well. So sometimes writing 2 emails to people actually helps, even 3, but make them different. Act like you're actually intelligent. Be original. It really helps to be original. And that really means if you hear a good idea in a lecture, it probably is no longer an original idea, because not only has the lecturer just spoken about it, but everyone else in the audience has also heard it. So try to think about something which relates to your background, something that you can do yourself. And the final bit of advice is a bit surprising. Be kind and good. And I tell people, this sounds like bleeding heart good. I say, even if you're really nasty inside, act like you're kind and good, so you'll get much more from people. So, okay. I wanted to just thank my towering heroes of science, Schneur Lifson, people from Cambridge, John Kendrew. He actually had left Cambridge by the time I got there, a year later. And my PhD supervisor is Bob Diamond, who's a crystallographer. I was, in fact, his only PhD student. Max Perutz, Francis Crick, and Aaron Klug, who also has a Nobel Prize and is alive. It's interesting that Bob Diamond and I, although he was my PhD supervisor and he actually supervised me, we never published together. We actually decided to publish 2 papers that would have adjacent page numbers in the same journal. But that was it. And this was not considered surprising. It was considered totally normal at that time. I've had an interesting group, about 1/3 PhD students and 2/3 postdocs, coming from all over the place. In the first 20 years of my career, I actually had nobody. And not until my eldest son was 18 did I dare take on supervision of a younger person. And that was actually quite good. Collaborating with colleagues actually worked very well. Many of my papers involve A and B or B and A. We often symmetrized it. But there were people who were at a similar level. And that was actually was quite a good idea. I do want to thank the Nobel Committee in Chemistry now. This is like, of course he wants to thank the Nobel Committee in Chemistry. You guys wouldn't be taking me out to dinner tonight. I wouldn't have to wear a suit and all this stuff. But in fact, they did something quite interesting. So firstly, for this audience, they gave a Nobel Prize that involved computers. And that is unusual. And although the prize was in chemistry, this was really for computers in biology. I mean, they call them complex molecules, but complex chemical molecules are biology. The second thing is they gave the prize for a method. I mean, it wasn't like we had solved something particular. And they told me that they expected to give more prizes in this area, and the idea was to get this early stuff out of the way so they could do so. And the third thing is they gave the prize for multi-scale work. Now, a Nobel Prize is meant to be for one thing. So we got it for one thing. It was called multi-scale. But by definition, multi-scale is more than one thing. And it's interesting that they were very proud about this. They said this was the key that allowed them to do this. They told me that multi-scale in Swedish is flerskalig, and that is a word that's hardly ever used in Swedish, would be more used now. So it was interesting. Okay. But of course, the field of computational biology and computing was the big winner. John Hennessy, a computer scientist, when there was the press conference, on the stage with me was him, the dean of the medical school, and the chair of computer science. The chair of our department was sitting in the audience planning his revenge and what he would have to extract from the president for doing this. And it actually worked quite well, because he felt this was a Nobel Prize for computing. But in fact, I think the big winner is the use of computers in biology. I think this is an area that has expanded enormously. But this is also sort of like a joke slide, because about maybe 10 days after the announcement, by chance I actually met the coach of the football team at Stanford. I'm not a football fan. I mean, I'd never seen football, but I met him, and that week Stanford had been accused of cheating in football by claiming timeouts when they shouldn't have got them, so I met the president. I mentioned this to him. In any case, I got invited to go to the football game with my wife and my grandson. So the field actually means the football field, and here am I on the Stanford 10-yard line, and here's my 10-year-old grandson at the time. I'm wearing a T-shirt that says Nerd Nation, which is Stanford's kind of like signature T-shirt. And although I guess on a football field, everybody, including the football players, is always very small, they have these huge TV screens up here, and basically everyone stood up and was screaming, "Nobel Prize, Nobel Prize." That was a serious high. So, and I just wanted to thank you all for your attention. You can ask me anything.
Martin Karplus, at the time of the awarding of the Nobel Prize, reminisced that when they were first getting around to publish the first publication of Charm, within the lab, they call it HARM, H-A-R-M-M, and they were about to publish this, and they said, no, this is not so very propitious for a computer name. We better hang the C for chemistry on in front, and they gave us Charm. 30 years later, Karplus reminisced that, well, maybe they should have left the name HARM as sort of an admonition to the harm to the garbage in, garbage out one can do with these programs. Any comment on that?
Well, I think that there's no doubt now. I mean, what's really happened is that many simulations are done by experimental teams, maybe too many. So therefore it's something which has been accepted. I think people now realize that molecules are very flexible. I actually believe that these calculations are really revealing useful things. They're best done in a very close association with an experimental team. I think, I don't see this as harm at all. Maybe Metropolis Monte Carlo should have been called harm, if you want, for obvious reasons. I think that it's a learning process. I think the most amazing thing about these simulations is that calculations that initially were done for 1,000 steps can now be done for 1,000 million steps. And the results don't suddenly, nothing bad really happens. I mean, good things, the behavior seems to be more and more sensitive. One of the troubles is that it's really hard to measure these things. I mean, in some senses, people are now doing single molecule work, and I think the gap is closing. But I honestly don't think that this has caused any harm. I'm not a carpus either.